So we have you guys for the last type of immunoassay which we'll be looking at is what is known as what the ELISA. So full meaning of ELISA is what? Enzyme-Linked Immunosorbent Assay. Enzyme-Linked Immunosorbent Assay. For the definition, we say that what the enzyme-linked immunosorbent assay is a widely used laboratory technique. Okay, so it's used for dictating and quantifying soluble substances such as what peptides, proteins, antibodies, hormones. Okay, so this ELISA actually what relies on specific binding of an antibody to its antigen. Okay, so meaning if an antibody is bound to something, that means the antigen is there, right? So it uses what enzyme that is linked to an antibody or antigen as a marker for dictation of the binding. Okay, so we say that the enzyme catalyzes what a reaction with a substrate to produce a measurable product using a color change. Okay, so for the types of ELISA, we have the direct one. For the mechanism, you say that the antigen is what immobilized on the microplate, and enzyme linked what primary antibody is what added directly. Okay, then we have the indirect one. This one what the antigen is immobilized on the microplate, so a primary antibody is added followed by an enzyme linked what secondary antibody that binds to the primary antibody. We have the sandwich ELISA. This one is that a capture antibody is immobilized on the plate. The antigen is added and binds to the capture antibody, followed by an enzyme-linked dictation antibody that binds to the antigen. Lots of English which you can actually pause to look at it. Okay. Then for competitive ELISA, we say that the antigen in the same competes with what a labeled antigen for binding to an antibody. Right. So procedure will say that a known antigen is what concentration of the antigen or antibodies absorbed on the microplate. So blocking, so non-specific binding sites are blocked using a blocking buffer. Incubation, the sample containing the target antibody or antigen is what added and incubated to allow binding. So washing, so that unbound substances are removed through multiple washing steps. Dictation, an enzyme-linked antibody specific to the target antigen or antibody is added. The substrate addition, a substrate is added and the enzyme catalyzes a reaction, resulting in a color change. Okay, so that's focusing on the types, which is direct, indirect, sandwich, competitive.